Hey, you are watching AliAziz YouTube channel.
Enzymes in Biochemistry. AliAzizQualityEducation YouTube channel provided you the medical related topics videos. So don't forget the subscription button. Enzymes are the unsung heroes of biochemistry, playing an indispensable role in the chemistry of life. Without enzymes, the biochemical reactions that sustain life would occur at a pace too slow to be compatible with the existence of living organisms. They act as biological catalysts, speeding up reactions and making the complex chemistry within our cells possible. Understanding enzymes not only reveals the intricacies of life but also opens up avenues for medical advances, biotechnology, and industrial applications. In this article, we explore the world of enzymes, what they are, how they work, and why they are so essential. What are enzymes? Enzymes are proteins that act as catalysts in biological systems. This means they increase the rate of chemical reactions without being consumed or altered in the process. Each enzyme is highly specific, typically catalyzing a single type of chemical reaction or acting on a specific substrate, the molecule upon which an enzyme acts. Enzymes are vital to every living organism, from the smallest bacteria to the most complex human systems. They drive the metabolic processes that sustain life, such as digestion, energy production, and DNA replication. The structure of enzymes. Enzymes are made up of long chains of amino acids, folded into unique three-dimensional shapes. The specific structure of an enzyme is crucial to its function because it determines the enzyme's active site. The active site is the region of the enzyme where the substrate binds, and the reaction takes place. This structure-function relationship is a key concept in enzyme biochemistry. Even slight changes in an enzyme's structure, due to mutations, temperature changes, or pH shifts, can disrupt its function. For example, high fever can denature, unfold, enzymes in the human body, leading to impaired function and, potentially, serious illness. How do enzymes work? The main role of enzymes is to lower the activation energy of a chemical reaction, which is the energy required to start the reaction. By lowering this barrier, enzymes allow reactions to occur more rapidly and at the relatively mild temperatures of biological systems. Enzyme action can be broken down into a few key steps. Substrate binding. The substrate molecule binds to the enzyme's active site, forming an enzyme-substrate complex. The binding is highly specific, often described by the lock and key model, where the enzyme, lock, perfectly fits the substrate, key. Alternatively, the induced fit model suggests that the enzyme changes shape slightly to accommodate the substrate. Catalyzes. Once the substrate is bound, the enzyme facilitates the conversion of the substrate into the product, S. This might involve breaking chemical bonds, forming new ones, or rearranging atoms. Release. After the reaction, the products are released, and the enzyme is free to bind with another substrate and repeat the process. Types of enzymes. Enzymes can be classified based on the type of reaction they catalyze. The 6 major classes of enzymes are oxidoreductases. These enzymes catalyze oxidation-reduction reactions, where electrons are transferred from one molecule to another. Examples include enzymes involved in cellular respiration, such as cytochrome oxidase. Transferases. These enzymes transfer a functional group, like a phosphate or methyl group, from one molecule to another. Kinases, which transfer phosphate groups, are an example. Hydrolases. These enzymes catalyze reactions that involve the cleavage of bonds by adding water, hydrolysis. Proteases, which break down proteins, are a well-known group of hydrolases. Lyases, these enzymes break chemical bonds by means other than hydrolysis or oxidation. They often form double bonds or add groups to double bonds. Isomerases, these enzymes catalyze the rearrangement of atoms within a molecule, forming isomers. Phosphoglucose isomerase, for instance, plays a role in glycolysis by converting glucose 6-phosphate to fructose 6-phosphate. Ligases, these enzymes catalyze the joining of two molecules, often with the use of energy from ATP. DNA ligase, used in DNA replication and repair, is a well-known example. The importance of enzymes in metabolism. Enzymes are at the center of metabolic processes, the complex web of chemical reactions that occur in cells to sustain life. Without enzymes, metabolism would grind to a halt. Some key examples of enzyme involvement in metabolism include glycolysis. Enzymes like hexokinase and pyruvate kinase facilitate the breakdown of glucose to produce energy in the form of ATP. DNA replication. Enzymes such as DNA polymerase are responsible for copying DNA before cell division. Detoxification. Liver enzymes, like cytochrome P450, help detoxify harmful substances in the body, converting them into less toxic forms. Enzyme regulation, controlling the pace of life. The activity of enzymes is tightly regulated in the body to ensure that metabolic processes occur at the right time and rate. There are several mechanisms by which enzyme activity is controlled. Allosteric regulation. Some enzymes have sites, other than the active site, where molecules can bind to increase or decrease their activity. This allows the cell to fine-tune enzyme activity in response to changing conditions. Covalent modification. Enzymes can be activated or deactivated by the addition of a chemical group, such as a phosphate. For example, the enzyme glycogen phosphorylase is activated by phosphorylation, helping to release glucose from glycogen stores. Inhibition. Enzymes can be inhibited by specific molecules. Competitive inhibitors bind to the active site, blocking the substrate, while non-competitive inhibitors bind elsewhere on the enzyme, changing its shape and reducing its activity. Many drugs work by inhibiting enzymes involved in disease pathways, such as statins, which inhibit the enzyme HMG co-reductase, lowering cholesterol levels.